Hello, everybody. Nice to have you attending this Seed Health Committee open meeting. I'm very glad that, well, many people registered and are attending. We are 50 people today. And I'm going to share my screen and show you the program. And after I will give speak to the presenters. Just for you to know that everybody is muted. This meeting will be recorded. When you have a question, we will do a question session after each presentation. So if you have questions during the presentation, write them in your question area on your screen, and I will ask the question at the end to the presenters. Presentation will be 15 to 10 minutes, and after we will have the questions. Okay, so now I'm going to. So I think it's the first Seed Health Committee open meeting that we really do fully online with nobody in the same room. It's a new thing, but what I'm very happy is that it allows us to have more people attending, and really I'm very happy to be able for us to give you some updates on all the projects we are working on. So I will do a short introduction about more generally the activities of the Seed Health Committee with the proficiency test and the training course and seminars that we are organizing. And after, we will go deeply in the method validation projects that we have under progress with Aurélie, who will do the presentation about the insects detection project. Isabelle will speak about Ascochytarab on chickpea, root on Fusarium on tomato. Guro will speak about Fusarium on cereals. Geoffrey will give an update on the project on Tilachia on wheat, and Ilaria will finish with botrytis on hemp. So just as a reminder to Terry, who left us and passed away last December, who was our vice chair. Here you have the members of the Seed Health Committee, and we are just needing now to elect a vice chair, and well, bye-bye, Terry. We have new members, some from 1918, Marianne, Isabelle, Koe, and Ruth. And we just elected a new member that who I'll welcome I'm very happy that we have Dr. Mahesh from India, who was elected this year, well, during the month. The proficiency tests which are planned, we have one on Pseudomonas syringae patovar PZ in 2021. Ruud is organizing it in collaboration with us. So the seed lots have been identified for with a medium infection, with naturally infected seed lots. We are very happy to find some. And we are also working on artificial contamination to obtain a high level. For Ditilencus dipsaci on alfalfa, it's going also to be organized this year by GVS. The seed lots have been identified for a medium infection, and we just need to check that the homogeneity is okay, and for the healthy level. And we will also, sorry, I have a little problem. Yeah. We will also do some artificial contamination for medium and homogeneous level. We will have one on Microdocium nivale. It's Marianne who is organizing it this year or next year. One on Paratastagonospora nodorum. Ilaria will organize it next year. And one on Coletotrichum lindemutianum. It's also being to be organized this year or next year by Kohe. And for all these proficiency tests, we really need infected seed lots for this proficiency test, and also for validation projects, mainly for Coletotrichum and Ascochytarab. We are really missing. and for targets for insects also. So if you have infected seeds, really feel free to contact me or Nadine. The training, the workshops, and the seminar, the CIDELS, we have a CIDELS symposium in Canada planned next August in 2022. It will be followed by a workshop on Qpcr for detection of Leptosphaeria maculans in rapeseed. So it's Stefan from Canada who is organizing it. With the CIDELS committee, we are planning an online training at the end of this year and in 2022, where we will speak about the general principles of CIDELS testing and seed-borne, seed-transmitted, questions about samples, subsamples, and quality assurance. And also what is important to pass an audit with all these quality assurance questions. For bacteria, we will work on dilution plating with Xanthomonas on bean, seed extract PCR with Xanthomonas on cabbage, grow out tests with Acidovorax on lamb lettuce. Viruses, we will work on ELISA for squash mosaic virus with bioassays with tobamoviruses. And for fungi, we will look at the methods with incubation on media or blotter, and the model will be Alternaria on carrot. Embryo extraction with Ustilago, and seed wash with Stilecia, like here. And for nematodes, we will work on Ditilencus and Amphilancoides. So it will give an overview of the different techniques used for seed health testing with some examples, and we plan to have some lectures and some demonstrations with little videos. Thank you. No, I don't see questions for me on this general presentation. Yes, I have one question. As an ISTA member, any laboratory may participate in proficiency tests. Yes, it is possible to participate to proficiency test as whatever laboratory it is. For accredited laboratories, participation to a proficiency test in seed health is compulsory, like for germination and purity. If you are accredited in one method in seed health, you have to participate. But if you are just an ISTA member accredited for germination and purity, but not for seed health, you can participate and you don't have a fee to pay because it's in your registration and membership. And after, for non-ISTA members, it's participation as a volunteer way. And in this case, for non-ISTA members, there is a fee. So, no, I don't see any other questions. So I hand over to Aurélie, who is going to present the project on insects.
Hello, everybody. So can you see my screen?
Yes, we can see it.
Okay. So hello, everybody. I'm Aurélie Charrier. I'm working at GBS in France. And today I'm going to present you a TICOM-ISTA project concerning exploration of methods for detecting insect in seedlots. Two committees are involved in this project, the Advanced Technology Committee and the CIDELS Committee. And the members of this project come from many countries, Denmark, France, the Netherlands, and Brazil. So why a project on insects? The insect pests are an increasing problem. The control of insects is becoming more and more complex, and the damage caused by insects are very significant impact, in particularly in terms of economy, environment, and food safety. The seeds can be an important vector for the spread of insect or disease, and many countries impose restriction for the importation of seed lots concerning the presence of insects. Currently, the ISA rules do not provide methods to respond to these demands. So the aim of this project is to explore different methods for detecting and monitoring the presence of insects in all their forms, larvae, nymph, or adult forms, outside and inside the seeds. The project is divided into 3 tasks. Task 1 concerns the identification of the needs of laboratories and stakeholders, and task 2 concerns a bibliographic research of existing methods. These 2 tasks will allow us to define the choice of seed-insect combination and the method that will be used in this project. The main task is the task 3, about the collection and the preparation of seed sample and the realization of the test. The project end is programmed in 2023, and a report will be produced and a workshop will be organized. So about the task 1, a survey was sent to ISTA members at the beginning of this year in order to identify the needs of the laboratories concerning these topics. In the table, you can see a summary of the results. 25 participants from 20 countries answered to the questionnaire. The main cyst species listed were some Fabaceae, Poaceae, all seeds and legumes. And the main insect species were Bruchus sp, Cytophilus granarius, Ligus sp, Ostrinia nubilalis, Tribolium sp, and Oryzaphilus. This survey also allowed us to identify the major difficulty for the laboratories, such as problem for identification, lack of training or methods, the insect diversity, and also the need of reference material. This survey also allowed us to list the use method already in the laboratory, field inspection, visual inspection, and also the use of traps. Concerning the task 2, it's a current statute overview. The member of the project made that at the beginning of the year. And we can say that insect pests may be divided into primary and secondary pests. Primary insects, like Cytophilus sp, attack whole and unbracket seeds, and secondary pests, like Tribolium sp, attack only damaged seed and dust. We produced literature research as a report, and this report will be sent to ISTA the next month. These 2 tasks allowed us to make the choice of the seed-insect combinations that will be used in the project. In the table, you can see the 4 combinations that we have selected. Faba bean, common bean, and lentils with Bruchus sp, cereal and rice with Cytophilus sp, Docus carota with Ligus sp, and corn with Ostrinia nubilalis. You can also see the method that we have listed in the project, like visual inspection, X-ray, oxygen consumption, barless funnel, spectral imaging, and sieving. The methodology that will be used is 1 sample for each method for 5 repetitions on infested and non-infested seeds. Now I'm going to do a quick overview of each method. For visual inspection, that will be made during the purity test by observing holes on seeds and insects in inert matter. But the disadvantage of this method is that this method doesn't detect insects inside the seeds. For the barrelless funnel, it's a trap. Sample or class in the barrel funnel under a lamp. Only insects fall in the beaker with alcohol. The efficiency of this method depends on the insect stage, the size, the seed size, and the humidity percentage. And also, this method doesn't allow to detect non-mobile stage of insects or the hidden forms. For sieving, it's an adaptation of a nematode detection method. It's allowed to separate the insect from seeds in order to detect outside forms and dead and non-mobile insects. But this method could be dependent to the size of the seed or the size of the insect. For X-ray, this technology allows to see inside the seeds. This technology is present in the ISTA rules in chapter 14, but could be improved. Currently, two existing technologies are available, 2D X-ray and 3D tomography. For 2D radiography, this method allows to detect the hidden form inside the seeds. The advantage is that is a non-destructive, fast and easy method, but we have some question about that. This method doesn't detect insect outside the seed, and we have question about the ability to detect damage in small seeds, and also to the ability to directly identify the insect species. For the 3D radiography, also called tomography, the advantage is that it's a very accurate method. This method is already used in many research programs, but we have also some question about that. Is it a non-destructive method? Is it enough to identify insects? And also this method is time-consuming and cost. The oxygen consumption is directly related to the insect respiration. Oxygen measurement instrument already exists. It's already used to help the efficiency of hermetic technology to protect stored products. So we have the question about the possibility to transfer this method to diagnostic and to the fact to detect and quantify insect on sample with this method, and also question about the identification of the insect. The last method is a multispectral imaging with a lot of advantage. It's a fast and accurate method to measure spectral and spatial characteristics. It's a non-destructive method and no need of preparation for the sample. But the disadvantage could be that this method can be not adapted for the hidden form of seeds of insects. So in this slide, you can see the timeline for the project. We already done the task 1 and 2, and the report will be sent to ISTA the next month. We just start the task 3 with the collection and the preparation of the sample, and the testing will be start in the next weeks. So a report and a workshop will be organized at the end of the project in the beginning of 2023. Thank you for your attention.
Thank you, Aurélie. Don't hesitate if you have questions. I had a general questions that I will answer at the end about proficiency tests. And for this question, please do not use the chat for your question, and you just use the question box. It's easier. I have a question. In this project, we are looking for insects, but what about insect eggs? Are these also included to be detected, since these are also likely infection cause? So do you plan in your project to also focus on detection of eggs?
Yeah, we perform experiments with different methods in order to detect the insect in all their forms. So it includes the eggs. So we think with the panel of methods that we have listed, we can find a method adapted for this form.
Okay, thank you. I had another question. Question from Dr. Sankaran from India. In insect damage assessment, in the lab, quantification is a problem. Any guidance? So apparently it's the problem could be the quantification of the insects inside the seed sample. And do you plan to work on this part in your project?
Yes, we not just detect, but also denumbrate the number of insects in the sample.
Yeah, and with various techniques.
Yeah.
Okay, and I don't see any other question. So and now it will be Isabelle who will present us the work she's doing on Ascochytarab on chickpea.
Do you see my screen?
No, we
Yes.
see your other screen, just the screen without the presentation.
It's
yes,
okay like
perfect.
this?
Yeah.
Okay. So I will present you the project for the detection method of Ascochytarab Rabiae on chickpea. It is a part of a French project. The name is Ascolup. It's a French project led by the Technical Institute in Ovia. GWS is involved in this project, and two pathogens are studied: Ascochytarab on chickpea and Coleotrichum lupinii on lupin. The aim of this project is to provide tools to producers for the management of this disease in seed production and in seed consumption too. Chip blight is one of the most serious diseases of chickpea, and this pathogen is selectively attacking chickpea, and it persists in the crop residues, seeds, and weeds. So GWS is involved in several tasks in this project, in the characterization of the genetic and phenotypic diversity of both pathogens, the validation of a detection method for Ascochytarab on chickpea, to develop a resistant test in controlled conditions as support of breeding, and to look for alternative seed treatments for lupin. So to focus on the detection method, is based on the test plan prepared by Siam Assad, member of the CDELS committee. And we need to have a quantitative method. The method is based on agar plating with a morphological identification, following by a pathogenicity test as an option. So the aim is to propose a new ISTA method for this pathogen. The test plan was submitted to ISTA reviewers and accepted. It was done according to ISTA guidelines for method validation, and it includes study of performance criteria of the method. We have all the seed material, infected seeds and healthy seeds too, and we set up a collection of target and non-targets. We have 20 isolates of targets coming from different areas in France, and 20 isolates of non-targets of other pathogens present on chickpea or saprophytes. We have already validated some performance criteria like analytical specificity. The morphological criteria was described for the target, and we compare the collection of isolates to the criteria. And to check if the targets, 20 isolates of targets meet the criteria, and if the non-targets do not meet the criteria. We done the characterization of the collection by the measure of spore size and growth, and on two media, Multi-Spectral and PDA. And the results of the analytical specificity was 100%. So this criteria was validated. But we have the other criteria to validate. The analytical sensitivity, and it will be validated if one infected seed is detected in 400 seeds in 10 replicates. The robustness will be done on different media by the variation of the duration of incubation, the light conditions too. For the accuracy, we will analyze one healthy sample, one low infected sample of 0.25% of infection, and one medium infected samples, about 5% of infection. We will do 3 replicates for each level of infections, and to evaluate the repeatability. And for the intra-laboratory reproducibility, we will perform this 2 times. And we will check the inter-laboratory reproducibility in a CT. This CT is planned for the first quarter of 2022. And we have currently 5 participants. So if you are interested to participate to this CT, don't hesitate to contact me. You will be very welcome. In the method, we have a pathogenicity test as an option. So to validate this pathogenicity test, we at the beginning compare 4 media for isolates multiplication, Multi-Spectral, PDA, V8, and CSMDA. And we chosen the CSMDA because it gives the best correlation for the Ascochytarab. We compare 3 pathogenicity test techniques: the inoculation by soaking germinated seeds, inoculation by deposit conidial suspension on germinated seeds on an agar medium, and we choose SCMDA, and inoculation by deposit the conidial suspension of spores on germinated seeds in potting soil. After the results of this comparison of 3 pathogenicity tests, we chosen the first, the inoculation by soaking germinated seeds, because we obtain more symptoms with these techniques and it's easy to implement. The second was rejected due to a high risk of contamination by saprophytes because we use a media. And the third was rejected too because we obtain less symptoms. So the performance criteria have to be validated. The analytical specificity, we will perform this by testing target and non-target isolates using the collection described at the beginning. For the analytical sensitivity, we'll use a dilution series on 10 seedlings. And we will choose the concentration fit for purpose, and it will correspond to the maximum of seedlings showing symptoms. For the robustness, we will check different parameters, temperature and light conditions. And for the accuracy will be tested at the same time as the analytical specificity on the isolate collection. And we will do 3 replicates for the repeatability and 2 times intra-laboratory to evaluate the reproducibility. And the inter-laboratory reproducibility will be performed in the comparative test planned, like I said before, the first quarter of 2022. It's cool. Thank you for your attention.
Thank you, Isabelle. Just to let time to people to ask you their question if they have, I had one general question. What we need to participate in collaboration with committees research, and I think it can be for your project, but also for all the other projects which are presented today. It's very easy. Either you contact the project leader of each project who is presenting today by email. Either you can contact me and I will forward your email to the one who is in charge of the project. And I have no other question for Isabelle, so I hand over to Ruud, who is going to speak about detection of Fusarium on tomato.
Hello, everybody. Is my screen visible, Valerie?
Yes.
It's perfect.
Perfect. Well, I will give you an, oh, one moment. Yes. We'll give you an update on the project which I'm leading, and that's also on a pathogen which is currently not a method yet. And this is identification of Fusarium oxysporum. So Lycopersicea, and which is pathogenic to tomatoes, and this is for screening tomato seeds, of course. Okay, how does it look like? It's a little bit of recap on what I presented last year, because this project is already running since, well, it was approved by the beginning of 2020, half 2020, and this is actually what we're trying to obtain. So it consists of media detection, and well, you can see at the right here there are the plates. So we plate the seeds out on agar, medium, growth medium, and let them grow. Let the fungi grow and see whether we can detect well, the Fusarium or not. And well, if we do not see any suspect colonies, then we can simply say that there's no Fusarium oxysporum present. And here it's bluntly stated Lycopersicea, not detected, but if there's no growth, we can most definitely say that there's no Fusarium oxysporum. Because well, the Lycopersicea is, well, Fusarium is quite difficult, or very difficult actually, to discriminate between different Phasio species on the morphological characteristics. But if we do have a photograph like this is in extremis, of course, with all seeds infected. But let's say that we have a lightly infected seed batch, then you occasionally will find these well, orange-like halos on this type of agar. And yeah, under a microscope you can see the nice, what is it, rod-shaped Fusarium shapes. And well, different from what Isabelle was talking earlier in her presentation on Chickpea, is that we do a TACMAN PCR identification in there, and it's optional, of course. So this scheme can be adapted a little bit, because you can choose whether you want to do a TACMAN PCR identification or a pathogenicity assay or both. But since we are preparing the whole method, I included it in this scheme, like in this manner. With the TechMem Qpcr identification, it's quite easy actually. You will take a loop of your suspect fungi, boil it, so it's a rough, crude DNA extraction, and then perform a TechMem Qpcr, which you can see on the right is a quantitative PCR, which every cycle you can follow in real time. And you can see here 3 different colors, which correspond to 3 different assays, and I will come back to that later. 2 of them specific to the Lycopersicety type, and 1 is an internal amplification control to detect whether there is fungi DNA present, yes or no. Well, if there is a negative result, you can stop your research and say that well, the Fusarium is present, but it's not the Lycopersicety. If there is a positive signal, then you should go to a pathogenicity assay, and that's an option, of course. And pathogenicity assay is a dipping assay, so you grow your tomato plants to what is it? Let's say 2 weeks or 3 weeks in size, and then dip the roots in a Fusarium suspension, and then grow them for another 3 to 4 weeks to see whether there's some detection of the pathogen, yes or no. So, well, this is already being in process, of course, and one of the validation criteria described in the accepted validation plan is the analytical specificity. So on media, there's 5 media that we choose as options for growing the Fusarium, and we selected 20 target isolates and 20 non-target isolates. This is a small summary of just the first 10 target isolates on the 5 different media. Corresponding to the media are also different temperatures, and also if they are growing in solidly in dark, or in first incubation a couple of days in darkness and then move to UV light, what is it, observations, or well, putting it on a UV light. And then afterwards, the growth was rated in fast growth, which is a coverage of over 50% of the plate. Slow growth, less than 50% coverage, and no growth of the isolates. And what you can see in the graph, in the table on the right, is that the mold agar MA and the PDO plus and PDA plus, which are quite close in preparation. There's only 1 deviance in an ingredient. There are fast growing ones, and Fusarium specific media and other media, the SNA, are slow growing or even no growing isolates are found with these medias. So overall, we can state that PDA plus and also the mold agar, which are easy with it, because you do not have to, you're not in need of a UV light. In 7 days, darkness 20 degrees, did show the best growth. Okay, then on the analytical specificity with the multiplex TechMem Qpcr. So I explained earlier in a presentation earlier that there are 6 genes, they called 6 secreted in xylem, genes which are contain specific regions which are only found for Fusarium, Oxyspora, Lycopersicety. And these are 6.2 and 6.5, and well, these are highly specific. And there's also an AF1A assay on an internal, that's an internal isolation control, which is an assay that reacts with DNA of all fungi. And here again, you see the 20 target isolates that we choose, and also the 20 non-target isolates that we've chosen. And well, they're coded, but I can tell you that they're all Fusarium oxysporum found on solanaceous, from solanaceous seeds or crops. And what we see there, when we use a threshold of 30 CQ, is that there's no false positive and no false negative results obtained with these 40 isolates. So that's what we want, of course. Then currently I'm working on the analytical sensitivity and selectivity. Well, like was said in the earlier presentation, we want to find one seed in 400 seed sample to be positive. Since we do not have positive, naturally positive seeds, that we did an artificial inoculation of the seeds and added one seed to 399 healthy seeds, and grow them on the 5 different media which were also used in the specificity part, to see whether we have only one positive result or more positive seeds popping up. We hope, of course, that we only find back the one and that the growth is good detectable. And we expect, of course, that the 2 previously good working media behave better than the other 3. So well, we plated 20 seeds per sub on a media plate. So for what is it, 400 seeds, it's quite a lot of work. I'm putting them all on the plates, and they are grown under the different growing criteria which correspond to the media. In this experiment, we also incorporated a selectivity part, and that's done by selecting 4 different tomato seed batches, which serve as a background seeds. So these are all 399 seeds are selected from a plum type, a cherry type, a beef type, and a cocktail type. And these are all spiked with this one seed and all put on these 5 different media. We hope to, well, we expect actually that there won't be any difference between the type of the seeds that we use, but yeah, you need to determine that, of course. Then the next steps, I showed in the flow diagram that a pathogenicity assay also needs to be incorporated, of course, in the validation. And there's within for DUS testing for new races of tomato species. There's a whole method described with morphological criteria, but also with resistance criteria. And there is a nice experiment described on how to conduct a pathogenicity assay for Fusarium oxysporum Qpcr, also called FOL. And so what we need to do is to perform the pathogenicity and validate that. I described it that all 20 full isolates and 20 non-full isolates will be integrated in this pathogenicity experiment, but that's something we, I think, have to discuss within the CTELF committee group, because it's a lot of work, and see if we definitely need this all. Next to that, of course, also described. Hey, there you are. Sorry, now I lost my presentation somehow. There we go. Yes. Next to that, of course, there's a proficiency test or a comparative test, I have to say, need to be organized, which we need 6 members for. I currently have 3 participants for the comparative test, so if you're interested, then please reach out. This will be organized, I think, in the end of next year. And yeah, with this comparative test, we also will integrate the reproducibility and repeatability, since we need to prepare, of course, the sets and also be participating. So that's an intra exercise for the repeatability, and all the participants performing the comparative test will perform it as described in the comparative test plan, and will act as reproducibility for the reproducibility characteristics. So that is actually what I wanted to tell. Thank you for listening to the presentation. And if you have any questions, please do reach out.
Thank you, Ruud. I had one question. Somebody asked, is pathogenicity necessary or could it be optional?
It is optional for you to conduct, but for the validation for ISTA, it's mandatory to have that in place. So that's why we integrated there. That's also something for the comparative test. If you want to participate, well, we want to have 6 participants which also do the pathogenicity assay, but if you're not willing to participate in a pathogenicity assay, please do reach out and just ask me or refer that you do not want to participate in the pathogenicity assay, but just in the part of the comparative test, since we need the participants.
And I think we also will be able to answer more easily to this question after all the validation process. If we see that it's 100% correlation between the TACMAN assay and the pathogenicity test, we can say that really we have a lot of chances that when we do only the TACMAN, we have our information is a Fusarium oxysporum from special Lycopersicii present or not in our seeds. The pathogenicity test would just be here to say, and it is pathogenic.
Yeah, and it's an option, of course, that, well, I can imagine that not all labs are able to perform a Qpcr or TACMAN PCR, so then the pathogenicity assay is still there for another option to indeed demonstrate the pathogenicity of the isolates.
I had another question. 20 seeds on a plate is a lot. Is there no saprophytes issues?
Well, of course, we see some saprophytes coming up, but since we're growing it for just a week, the growth rate is not so big that we see difficulties in this validation. But I can imagine that if you have a mucor on it, then it will rage through your plate and then there's no detection possible. Although with tomatoes, I do not see that happening, because tomato seeds are quite protected by the fruit itself, of course. And also some kind of treatment is done while harvesting or after cleaning the seeds, actually. So if the fungi is present, I do think that it will come from the inside of the seeds.
Another question, in seed health tests, for comparing between lab results, do we have tolerance limits tables as we have for purity and germination results comparison? I don't know if you feel okay to answer.
Well, we have, of course, our seed health PT documents, right? And there's a whole calculation screen that you can also use for your comparative test. So I think that's present, but correct me if I'm wrong, Valerie.
Yes, that's true. We have our guidelines that you can all find on ISTA website in all the documents for method validation and proficiency test organization also. But that's true that we don't really have the tolerance tables like we have in germination and purity. For a result in seed health, it's generally detected, not detected. For fungi, you have a percentage of seeds, but we don't very often use the tolerance limits. So that's all the questions I had. I'm just looking for you. Yes, it's all. So now I hand over to Guru, who is, we are staying in the fusarium, but this time it's on cereals.
Yeah. And now should, yes, and.
Can you see the screen? Or
No, I cannot see it now.
take. Yes. Now, why should take few seconds?
No, I don't see it.
It's all dark, Andrea.
How it is?
Perhaps I could try to do it myself. You open up that window again.
Yes, now we see it.
It's there, and if you use the full screen, the presentation mode.
Yes, I just don't see this option anymore. Strange.
No, it's not in.
Okay. A second, I can try to open again.
It's
this one, yeah.
Now?
Yeah, there it is. Okay. Hello, everybody. It's going to be a bit more about Fusarium. Now it's about validation of detection and identification of Fusarium species in cereal seeds. And there are a number of species, and it's also 3 different cereal species. Here is an outline of my presentation, a few words about the background, objective of the study, pathogen list we are dealing with, main steps of the method, and validation procedure, comparative tests, just in general, some challenges that we have that we are struggling with, and then the project group and the timeline. Project. Next, please. Yeah, Fusarium and Microdocium are important seed-borne pathogens in cereals, and they can cause reduced germination, seedling blights, difficulties in establishment of a plant stand in the field. However, also Fusarium species cause Fusarium head blight, and some of the Fusarium species often develop mycotoxins in the grain, and because of that, Fusarium head blight has become one of the most important diseases in wheat worldwide during the last, let's say, 20-30 years. And that is, of course, due to the mycotoxin issue, because mycotoxins are toxic to people and to animals, and there are limitations for what is allowed in the grain of these of several of the toxins produced by some of the species, Fusarium species. And yeah, seed health testing methods for these fungi was developed by ISTA, Plant Disease Committee, as it was called at that time during the 50s and 60s. It was based on symptoms and morphological characters by the pathogens. A number of comparative tests was carried out, but the methods were not really validated. It is described in some of the old seed health testing handbook, but it's outdated or not available any longer. And but still, these methods are more or less in use in routines in some laboratories, and on either on agar plates, as you see on one picture there, which is represented both by Microdocium, Fusarium, and also some saprophytes. The other picture with oat seeds, you can clearly see brown discoloration of roots, and those plants will not develop proper plants in the field. And this is in oats, and Fusarium head blight or Fusarium species and damage has been a major issue in Norwegian oat production for 15-20 years. That is my motivation to take part of this in this project. Next, please. However, a seed testing method for Microdocium in wheat was approved by ISTA in 2007. It's method number 7022. It's based on morphological characters on agar medium. However, it is also a need for detection and identification of Fusarium species in cereal seeds, partly because of the increased occurrences of some of the Fusarium species due to the mycotoxin issues, and then it has also been requested by the seed industry to be able to perhaps avoid using seedlots that carry these unwanted Fusarium species. So on this background, a validation study was initiated some years ago, in fact. Several reasons for the delay. And now we want to detect both Fusarium and Microdocium in cereals, and it will include wheat, oats, and barley, and it's based on the Microdocium method in wheat. It's more or less taken for granted that new methods or method development nowadays should include or should be based on molecular tests or molecular techniques, but we experience that there is many primer sets and techniques for PCR identification of each of the Fusarium species. And given the main request or purpose of the method, which might be rapid routine screening of seedlots to assess the suitability and or need for fungicide treatment, we had a lot of discussion on that and did quite some work. But after, we concluded that PCR identification could not be included in this study. However, it will be looked at in a new separate project with new technology such as barcoding, etc. Next, please. So the objective of this validation study is to provide an internationally accepted method for detection of Fusarium also in wheat, barley, and oats. And this will enable then the laboratories that are recording Fusarium in addition to Microdocium when they are using this 7022 method to also be accredited for Fusarium, because it is used in routine screening in some laboratories, and they would very much like to have the method accredited. Next, please. Here is a list of the main Fusarium species that we are looking at. Species and Fusarium names is a complicated issue. The taxonomy of Fusarium has been confusing and differed through the years. So we have also had discussions on what is the name of the different species. And besides, the importance and severity differs also among Fusarium and Microdocium species. So on the list here, it starts with Microdocium, the two Microdocium species. They both cause damage and severe germination problems. And also Fusarium graminearum and Culmorum. And also perhaps Croculense, we are not that familiar with that one. And it's perhaps synonym with Cerealis or perhaps not. I don't know if we really have concluded on that. But in addition, also Fusarium avanasum, Fusarium poa are present commonly on cereal seeds. Then we have Fusarium lancettia, which is particular prevalent in oats, at least in the Nordic countries. And we also sometimes see it in symptoms, Protrichoides, Pseudograminearum. They are identified on cereal seeds, but the role of seed-borne inoculum and the damage to germination are not reported as far as we know. Next, please. So the main steps of a method is that seeds will be plated on PDA or malt agar, disinfected and plated on media in petri dishes, and based on the procedure described in the microdocument method. Incubation will be for 7 days at 20 degrees, either in dark or followed by some few hours in daylight or under near ultraviolet light, or under alternating 12 hours darkness, 12 hour NUV during the incubation. And it is then based on morphological identification, which can be not so easy. And so it includes also optional species identification on special agar types to have the species to have Fusarium producing spores, so it's more easy to identify to species level, like carnation leaf agar and synthetic nutrient poor agar. It depends, of course, also on the need to identify at the species level what the lab will need to do. Next, please. And this is showing, this is a workflow showing more or less the same as I said, that if we detect, we look at media and do we find any suspect colonies or not? If we find them, we try to morphologically identify. Perhaps it is not Fusarium, so then we didn't have any Fusarium in that seed lot, or it has to be looked at with the special media, and perhaps Fusarium species target is identified. And of course, those laboratories who have the equipment and possibilities to do, for instance, a PCR test, that could be useful to confirm what we find but this is more the workflow even when a seed lot is being tested. Next, please. Yeah, and we are then going through a validation procedure according to the ISTA guidelines and performance criteria like analytical specificity. We have been working on that so far. Results are under evaluation, and we see that it's not easy to get the same, the colonies look the same, even if we started with the same isolates, to get to look like the same in different laboratories. And then we are analytical sensitivity with spiking healthy seeds with artificially contaminated seeds, selectivity on different cereal seed species, robustness, variation of parameters, incubation, media, etc., and repeatability, reproducibility based on results from comparative tests, and then statistical analysis. I don't go into detail on that. Next, please. In the comparative test, which we are planning now, will be performed by using naturally infected seed samples with different levels of infection containing at least one of the Fusarium species in each cereal species. And it's necessary to do a homogeneity test before and a stability test after by the test organizer. That we in Norway are responsible for that. And each participating laboratory will receive 9 coded samples of 400 seeds, replicates of 3 seed lots, healthy, low, medium, and high infection of either wheat, barley, or oats. And we will also send out coded isolates for the different target species Next, please. As I mentioned, we have some challenges. Morphological criteria of Fusarium are not always conclusive, and despite descriptions based on recognized, renowned laboratory manuals, we struggle to obtain uniformity. And here is an example that, for instance, Fusarium lancetia, here is 3 different isolates looking quite different. Next, please. At the end, Seed Health Committee will discuss and decide if the outcome will be a new separate method for Fusarium or an updated version of the microdocument method, including also Fusarium in wheat, barley, and oats. Next, please. And so far, the project group consists of people from Human Seed Laboratory in Norway, NIBIO in Norway, from Italy, from Scotland and France. And here is an overview of what we are doing. Is the time more or less out now, Valerie? Does it, since I don't know. Okay, it's at least we shared the work in a way between each laboratory, because there are many isolate or Fusarium species to deal with. And for instance, analytical specificity, we do 1/3 of the isolates on PDA, some do it on MA, and some with SNA or the CLA to identify the species. Okay, next. And the test organizers, a lot of work has been done by Isabel in Cheves by collecting isolates and also working on the performance criteria, Isabel and her co-workers, I believe. And the comparative test will be organized from NIBIO and Human Seed Laboratory in Norway. I think that was last slide, wasn't it? Okay, just a timeline then. What we have done is develop the test plan, and it has been revised and sent to ISTA again. The isolation collection is done. Study of performance criteria from isolate specificity test is under evaluation, and the comparative tests are under planning. And yeah, we will hopefully finish within 1.5 years from now. Thank you. Yeah.
Thank you, Guru.
Next slide is one. Yeah, no, we stopped that. That is okay.
Thank you, Guru. I have one question, but it's more general one. How many labs are accredited by ISTA? The number of labs accredited in total, I don't know it completely. Andrea, maybe you can help me. It's 200 and something?
One sec. Yes, it's also, even I don't know the exact number of the laboratories accredited.
Around 200, let's say. But
yes.
for Seed Health, it's 29 laboratories, not many labs. Many labs are performing Seed Health, but are not accredited for Seed Health.
Yeah, and that depends. Different methods, I think. Where accreditation does not cover all kind of methods.
Exactly, yeah. And I had another question from India. Seed Health testing is quite complex and demands much greater skill and facilities for identification assessments and routine, but germination, for example, test or purity. Labs find it difficult to do perfect Seed Health test as a routine. So is it possible for ISTA to initiate a system for identifying, strengthening one or more of the accredited labs in the country to be the dependable centers for Seed Health testing in that country? Well, I don't think I can answer this question. I will forward it to the ISTA Secretariat and Executive Committee, maybe. But I recognize that yes, seed health testing is demanding quite some equipment, and it's an expertise which is pest by pest, not only general. So I can understand that it's more difficult to implement in a lab. But don't forget that the seed health committee is also here to help you. Well, at the moment it's not possible, but in the future, and to do some workshops in a particular area. If you want to implement some seed health testing, it's always possible to have some training with workshops. Why deep freezing method is not considered?
Most of all because it's much more work to do the examination, examination of the samples. It's much more time consuming, and you need to do a lot more of microscopy slides to look what you have there when this because there is often a lot of hypha and not that much Paradoxa from Fusarium, for instance. So we used to do that in Norway. We used the freezing blotter method for almost 15, 20 years in routines, but then changed to the agar plates after a workshop we had, where Valerie Cockrell was demonstrating agar method, and at least we changed to the agar plates. And then we also changed to the normal blotter test and just see the symptoms for in oats and barley. But wheat is testing on agar with the microdocker method, but Fusarium is also recorded. So, but freezing blotter is more, what do you call it, time consuming?
Time consuming.
Yeah, it takes more labor.
Yeah. Yeah, and we had compared it for FOMA on corn salad with another group. And we also saw that it was more difficult to harmonize and to validate because the light conditions can vary, and it can increase variability in results.
Yeah, we also did a lot of comparative testing in Nordic countries some years ago, and yeah, that was concluded to use agar plates.
So I have a question, and I think it's in Portuguese, if I understand it well, which is asking if it's possible to translate and to have an audio translation. I'm not so sure for today, but the presentation, well, all the meeting is recorded, so you can listen to it very quietly afterwards when it will be on the YouTube channel of ISTA. And I have no more question. So now I can hand over to Geoffrey for Tilesia. Geoffrey?
Yes.
No, it's okay now. Now it's okay.
Yeah, looks good now.
It's okay now? Okay. So my talk today concerns validation of a detection method on Tilesia on cereal seeds. So just before starting, I just want to thanks my trainee of this year, so Mathilde, that we have done a part of this work. So thanks for this, and yeah, let me stop now. Oh, okay. So just to introduce the disease and the pathogen. So Tilesia is responsible of burnt So it's a re-emerging disease since 2007, and it have a different impact, but we know that can have significant losses, especially on the yield. So it can have a very important impact in fields. So so the seed infected are not usable in mailing due to smelling rotten fish. We know that the certification is different between the countries, so we have various detection thresholds. So, for example, in France, we have zero. So the norm is at 0 spores per seed, when in Germany is 20 spores per seed, and in Australia 10 spores per seed. So it's really different between the country. So the performance of the method could be various also between the country. So currently in conventional seed, when Tillecia is detected, we applied treatment. So this is not in accordance with the reduction of chemical product. So it's really important to be able to detect the seed health quality in seeds before applied any treatment. So here we have some generalities about the method used. So most of the time, the method is a seed washing. So the seed is put in a buffer, and then after soaking and shaking, we can apply different filtration steps. So we have various filtrations. So some of them is to eliminate impurities in the seedlots, and some of them is used to collect the teliospores. After the collect of the spores, we can observe the presence of Tillecia on the microscope in order to determine number range of spores. And this allows to identify the different Tillecia species and also determine the concentration of the contamination. So in order to know which kind of method is used in the different laboratories in Europe or in the world, we wrote a survey, and this survey was sent to different labs by the ISTA or IPO networks. And by this way, we wanted to collect most information about the different protocols used. So the aim after the survey is to compare the technical variation step in order to harmonize the method, and of course validate the method based on the soaking and filtration to detect Tillecia in seeds. So here we have some synthesis resume of the result of the survey. So 14 participants completed the survey for all of them. So 100%, every laboratory works to detect Tillecia caries, whereas 65% works on Tillecia levis, and 70% works on Contreras. Only a few of the laboratory works on Tillecia indica, and this Tillecia is the only one Tillecia quarantine test. So we can see that the different participants came from different countries. Most of them are came from the Europe. So we have Germany, Scotland, Czech Republic, Estonia, Sweden, United Kingdom, Lithuania, Austria, Germany, also Germany, Italy, and France also. And one of them came from Canada. Everybody using washing methods, so in all the case, but the washing method was sometimes using filtration steps, so filtration materials in 65% of the case, and most of them using an internal protocol. So this means that the protocol were adapted from an official one, so in 70% of the case. So we have recorded a lot of variation of technical condition around the protocol, and not everybody use a PCR confirmation. Depending on the method, some of them can confirm the identity of Tilesia by morphological criteria. So after complete and collect all this information, we define a test plan in order to compare all the technical variation steps. So you can see here that we have a variation on buffer, shaking, filtration, centrifugation, and also reading. And in details, we can have variation on volume used for extract the spores, but also on the temperature of the buffer used. Some lab use room temperatures and other use heat buffer at between 55 degrees and 65 degrees. We also have a lot of variation on shaking step due to the time and speed used. We can see that we can have sometimes just a shaking by hand, or sometimes with a agitation table with different RPM. And the time also can be different with 2, 5, 10, and 30 minutes. The filtration is also quite different between the lab. The type of the filters is could be nylon or nitrate cellulose, and the size, of course, of the filter is also quite different, with 20 micrometers, 10 micrometers, 3 micrometers, but we have also found 8 micrometers used. So we have a lot of variation on this step. The centrifugation step is a bit particular because this step is used only when we used nylon filters, because in case of using nitrate cellulose, all the spores are collected directly in the filter, and the observation are done directly in the filters. Whereas when we use nylon filters, we can after that rinse the filter and centrifugate in order to eliminate supernatant. So the unit also of the results could be different between the labs. Some of them are explained their result in spore per milliliters, or in spore per gram of seeds, or in spore per seeds also. So we have many variabilities, and we defined priorities in order to compare all of them. So here we can find some first results. So the first step tested was the temperature of the buffer. So we compare on 3 different seed lots. So one naturally low infected and 2 other seed lots artificially infected, one in low infection level and another one with a medium infection level. So each condition were repeated 3 times, and we for each condition, we compare room temperatures buffer and 55 degrees temperature buffer and 65 temperature buffer. So it's not 1, 2, 3 in the histogram here, in the right side of the slide, and we can see that the results show more heterogeneous counting of the spore, heterogeneous in concentration of Tilesia when we used hot buffer, for example, in the low naturally infected seed lots. Here we can see that the scale is very high. And one of the steps confirms that a room temperature can be able to extract more spore, for example, for the artificial infected seed lots. So due to a longer, due to no hard step on this protocol, we decided to use room temperature buffer for the next of the experiment. Another first result concerns shaking. So currently we test only 2 seedlots, the low infected and the medium infected seedlots. And we can see that we compared different time of speed. So like I said, 2, 5, 10, and 30 minutes, but also a shaking by hand and also with an agitation table at 2 different speeds. So 150 RPM and 200 RPM. And the results show, I mean, they prove that when we have a shaking step, I mean by hand or by a machine, we have most of the time same results or similar results. I mean, this allows to extract spore from the seeds. And in one modality, we find a higher concentration of Tilesia. So if we are doing a statistical analysis on it, we can see that this modality, so 10 minutes at 150 RPM per minute, allow to have better results. But we need to confirm this first result with a high infected seedlots. But for me, the most important information that we can see that after a shaking, we can be able to extract the spores in similar quantities. So I don't know if we will find modalities, a best condition compared to the other one, but we can see that this one, I mean the 10 minutes one, have better result. So we will see in the next step if it will be confirmed. So the next step planned concerns we will also evaluate the volume. So we will determine a ratio of a buffer per seeds, so in milliliters per seed or per gram of seeds. And then we also compared so different type and size of filters. So these works were already done, and the result need to be discussed with all the consortium. But I take the liberty to don't present it here because we need to confirm the first observation, but it's ongoing about the filtration. And the other step will be done and planned to the end of August. So for the end of the training, we would like to restitute the first result obtained to the consortium, so to all the partners that are interesting for the ring test, and try to find to determine the best condition and to select the best condition together. So then the next step will be to evaluate performance criteria of the method. So we will are doing analytical sensitivity and specificity, but also evaluate the diagnostic performance, so specificity and sensitivity diagnostic, but also testing the selectivity, and of course when we will organize the validation ring test, evaluate repeatability and reproducibility. So another information was sent by Hippo a few weeks ago. So now we have 17 participants. So 3 of them did not complete the survey, but we discussed about which protocols they used, and it's quite similar that with the other conditions that we defined previously. And yeah, at the end, a validation report will be submitted to the for the HISTA groups. So currently the test plan for the comparative test is on review to the CDELS committee. So I think it's my last slide, and thanks for your attention.
Thank you, Geoffrey. I have some questions. One from India. Tilescia in wheat is a problem in India. Sufficient experience with Tilescia is there in India. Is it possible to associate Indian expertise lab to the project? It may add value to the HISTA's efforts in this regard.
I think yes. I'm still open to add a participant, and it could be interesting to add an Indian participant. So yes, but I think that the problem, the Tilescia problems include also Tilescia indica in India. Is it true?
I don't see an answer yet.
Okay.
Because yes, yeah. This validation plan will be all the Tilescias except indica. Yeah. But what I suggest is that you just contact Geoffrey if you want to participate to this validation study. As for the other studies, and if you don't succeed to contact the project leads because you don't have the email address, do not hesitate to contact me, and I will forward to each one.
Yeah. Okay. Because just a precision, like you said, the validation ring test will be organized with Tilescia seedlot infected by Tilescia, but without Tilescia indica, due to facilitate the sending of seedlots around the country, etc.
Okay, I have another comment. Tilescia is included in the Indian seed certification schemes, both for field and seed stages. And after, I have another question. Do you check the homogeneity of the Tilescia contaminated seedlots? Do you think 3 replications for each test is sufficient?
Yeah, well, yes, I don't present the homogeneity result of each seedlot, but each of them were tested 10 times, so in 10 replicates in order to check their homogeneity. And that's the reason why we decide to test only 2 seedlots for the comparison of the agitation steps due to an heterogeneity of the results on the natural one. So the homogeneity, yes, were checked before.
Another question, what's the correlation in Tilescia incidence between field and seed stages?
We have some results from other projects. So the question is, are we sure that when we are presence of Tilescia, we have an impact in the field? That's right. So one parameter that it's not taken in account here is the viability of the spore. And we have proved in other projects that with a very low infection level in the seedlots, I mean less than 10 spores, if the weather and if we are good condition of transmission, the pathogen can be involved symptom expression in the field on here. So in a very low infection level, we can have transmission. So yeah. But the most important information is the viability of the spores.
And that's all the questions I had for Tilescia. I keep some for the end because they are very general. Okay, thank you, Geoffrey.
Thank you.
And now I hand over to the last presentation made by Ilaria.
Okay, thank you, Verity. Can you hear me?
Yes, perfect.
Okay. Okay, thank you very much. Good afternoon or good morning or good evening to everybody. Well, I have the pleasure to introduce this vista test plan for the validation of the detection and identification of Botrytis cinerea on hemp seed. Next. Can you change the slide? Okay. The organization of this test plan is managed by GV and CREA. And the comparative test, which is scheduled by the end of the year, if we are able to hand the validation of the performance test, is always managed by GV and CREA in collaboration with the HempPeat company. And I just already anticipate you that 12 laboratories will be involved in the comparative test. Next. Before describing the test plan, I would like to talk about the origin of this test plan, of this method. Everything is with the new interest of hemp cultivation and consequently in the increase of seed production. This new interest is could be linked to, for example, to the development of hemp use for the production of sustainable building materials, thinking about the fiber for thermal insulation, or as a source of nutraceutical food, thinking hemp oil, and also for medical and recreational purposes. I talk about the extraction of cannabidiol, or CBD, for example, from hemp flowers. And that is very important. This is a very important new business in the last few years. Okay. Botrytis cinerea causes very serious losses in more than 200 crops species all around the world. And it is a fungus that is typically exudative on mature and sentient species. But it's very important that it is able to enter in the early stage of crop development, stay quiescent for a long period, and then rapidly rot in the tissues when change the climate and above all the host physiology. Okay. I briefly list the common symptoms because it's very important to remember this. Very common is the presence of this sore throat with a collapse and the water soaking of the parenchyma tissues, with the appearance of gray mycelia with a huge number of conidia. These symptoms are very typical on leaves and soft fruit. Very common are also the infection on senescent flowers, and that is the so-called blossom hand rot, and the subsequent spread of these rots in the developing fruits. But very important is the reduction of germination when botrytis is seed-borne. This presence is the presence of botrytis on seed is often combined with symptoms of damping cough and cell rotting at the ground level. And it's also important to remember that more than 50 species are interested by the presence of botrytis. Okay? The objective of this test plan is clearly the development of a protocol for the detection and for the identification of Botrytis cinerea on hemp seeds. Okay, the method is kind of a, please you can change the slide. I would like to say that the testing method is hand validation, and the structure that you can see in this slide. First of all, is a blotter method. We use a double layer of Workman paper number 1 or the equivalent. It's not a problem, but it's important that is the same here. We use petri dishes of 90 millimeters, and we put 10 seeds per plate. The pre-treatment is not a request, and the incubation is carried out at 20 degrees in the dark, and after 7 days, the botrytis is checked by naked eye and by microscope. Okay. As you know, to validate a protocol, we start in the validation method, and above all, we are now verifying the performance criteria. The first criteria I talk to you is the analytical specificity. That is the ability of a method to distinguish between the target organism, that is our pathogen, and from the other organisms. We are collected 20 isolates, 20 strain of Botrytis cinerea, that is our target organism, and 18 isolates of non-target organisms like Alternaria, Stenphilium, Epicoccum, Fusarium, Trichothecium, Penicillium, Aspergillus. That are these species are very common on NFCs. To verify the specificity, the strain are plated on Malta media and are also used to inoculate some samples of 10 samples of 100 seeds that was previously seeds infected, and these infected seeds are placed on blot to proceed with the exam of morphological parameters. Okay. In the next slide, I report by example the morphological criteria we use to identify botrytis on a blot. By naked eye, which search for the presence of gray mycelium with the sporulation. It's important that there be sporulation, because the soft growth can also be due by saprophyte. Thinking about Alternaria, that is very common on hand. After this, it's important to check the presence by microscope of like IFE, that's also very common and typical of botrytis, and the presence of a branching conidiophora. Then we control the size of conidia, the microconidia that are produced by conidiophora. On medium, we check the presence in the hedge culture of sclerotia, and we need to find this typical aspect of the fungus on the media. Okay. In the next slide, you can see 3 common saprophytes on M-seed in blot, which we commonly find using blotter for analysis. This is Aspergillus, Trichothecium, and Alternaria. And the analysis of this morphological criteria as the main topic for verify the specificity of the method. Now we are control and we are check the results of this first trial. In the other slide, you can see the study. Okay, you can change the study of the analytical sensitivity. That's please the next slide. Okay, the analysis of sensitivity. The sensitivity defines the limit of detection. This is the lowest quantity of a pathogen that can be detected in an analytical method. We are verifying these parameters by speaking of contaminated seeds by botrytis in a healthy seed sample. In particular, we work with a sample size of 400 seeds, and one contaminated seed is picked during the plating in a sample of 399 healthy seeds. And the last criteria, you can change again the slide, and I just end my presentation, is the robustness, that is the ability to vary according to small variation of parameters. We change the parameters of the method. We are evaluating whether the alternation of light and dark, 12 hours of dark and 12 hours of light, and the duration of incubation. We will do the notation at 5 days and 9 days instead of 7 days, and we check the control if these two variation of parameters have some effects in the detection and above all in the quantification of botrytis cinerea in naturally infected seed sample. In fact, we use for this robustness in naturally infected seed with an infection that between 4 to 5% is the best way to work for this evaluation. Well, I like to remember that these performance criteria are under evaluation, and we are trying to do the verify by statistical analysis, and then I would like to conclude and close with the presentation by showing you, if possible, some photos of the research center of Rovigo, where we work. Part of our activities is dedicated to research, laboratory research and experimental trials on that. Okay, I just.
Thank you, Ilaria. Your presentation has disappeared at the end.
I thought it maybe no. I thought it's another co-organizer. Now you can see it again?
Yes, perfect.
Okay. No, this is a picture of our field trial that we have in Rovigo, because we have a trial for breeding purpose, and this is a trial for where we can find several lines for medical and nutraceutical purpose, and this is a field trial where we collect where we have collected the seed to check the presence of botrytis and others are provided for this test plan. Okay, only this.
Thank you, Ilaria. I don't see questions from you. I have one. We were speaking for the Fusarium on cereals, and we had a question about why not deep freezing and why not a method on blotter. So what is the choice here between blotter or agar?
Sorry, I don't understand because I don't hear well. The connection is not very good in this moment. You can repeat the question.
Why is the choice of blotter or agar, and in this case blotter, where for example for Fusarium and cereals the agar was chosen?
Because botrytis is a typical saprophyte. It is common to find on the parts of the coat of the seeds. And remember that hemp is not a seed, is a arachenia. Arachenia is a fruit. And for this kind of seeds, it's better to use blotter because it's easy to check the botrytis. Thinking about, for example, botrytis on sunflower, we use the same way, the same method. Is better, and I check. I had some trials using PDR and blotter, and it's easy to find botrytis on blotter than on agar, because on agar I have a lot of saprophytes that go up the botrytis.
Okay, thank you. So I don't have any specific questions, so I'm going to ask all the panelists to switch on their cameras, not your microphone. If you can just switch off your microphone and switch it on only when you want to answer a question. I had several general questions. I go back to the beginning of the question. One of the questions was in proficiency tests for seed health, how do we take care of the country's PQ regulation compliance by the participating labs? Such PT samples may not be cleared by the country's PQ department. So I don't know for some of you if you have some examples about proficiency tests and sending, or if you want me to answer. Okay, well, I'm going to answer. In fact, when we organize proficiency tests, we always ask the participants if they need a permit and to send us the documents that they need for a permit. So in Europe, for example, if it's tomato, it can be a letter of authorization. If it's infected by a quarantine pest. It can be just a plant passport if it's not infected by anything that we don't want. For the US, for example, we have some APHIS permits that we need to send. So it really depends on the regulation of the country. Really, we work in close collaboration with the participants to know what is needed. But that's true that sometimes we have the impression that we have done everything and we sent all the documents which were necessary, and it's still blocked at the customs. So that's a real problem. And Andreas, the ISTA Secretary General, in another meeting advised in this case to ask your voting delegate in your country so that this person could go and speak to the NPPOs, and also the ISTA Secretariat can help to discuss with the NPPOs to explain our actions and our proficiency tests, which are very, very important, of course, for the labs to know how to work and to have good good base for their results. So yes, just contact your voting delegate if you have problems, and like that, we hope that we will improve the situation. So one of the questions. Sorry?
Can I add something to that?
Yes, of course.
Well, lately I was organizing a PT or a comparative test with a carotene pest. What I noticed there that there are extra labels and LOAs, of course, a letter of authorities needed. But what also struck me is that at customs you need to have a sort of approval from the local purchase importer, something like that, to deal with the costs. So predefine the costs that the package sending will cost, and decide with the participant of the country who is the one that needs to pay for these costs. It's about $150, I think, that you need to pay for an import.
Ah, okay.
So it's something worthwhile to mention as well.
Yes, that's something to know, yeah. And another general question I started to answer it was what's the need to participate in collaboration with committees research? Just contact the project lead for the project you are interested in, and well, generally to participate to a project for the validation part, we ask labs to be expertised in this technique or in this best, of course, but it's always possible to participate to some parts of the project. So you can contact the project leads and ask them and tell them also what you are working on. That's done. I had a comment saying, please provide CDL's testing and detection training. So yes, we are trying. Just feel free to contact us to ask what you need. I remember that in the past years, some countries asked us to come. It was the case in India. One company organized a workshop, and we as lecturers came and we organized that with them, just to train people exactly on the pests they wanted to be trained. So just do not hesitate to contact us if you need the CDL's committee to help to organize a training and a workshop. Are there any ISTA member laboratories accredited for pathogenic fungi in forestry seeds? I really don't know. What you can do is go on the ISTA website under accredited laboratories, and you can search by type of methods, and then you can click on the name of the laboratory and you can see all the methods they are accredited for. But I'm not sure there are many, if there are. I don't know any laboratory who is accredited for forestry seeds. You neither, Guru, because you are more working on forestry seeds. You're on mute.
You're unmuted. Yes, can I hear you? Can you hear me now?
Yes.
You hear what yeah. I don't know if there is any labs that are accredited for diseases on forestry seeds. No.
No, okay. Thank you. Yeah, it's a very rare thing for us.
Yeah.
And I had the last question, polymer, no, sorry. Polymer coating of seeds is becoming common in the seed industry. For CDL's testing, seed should be free from polymer coating. Is it not? Please guide. Yeah, that's really an issue we have in the CDL's committee, and we had many discussions. It's in fact very difficult to validate a method for every type of polymer treating, of coating of seeds or of treatments. So when we validate a method, we cannot test all of that. I don't remember when it was. It was in India or in a few years ago in one of the ISTA meeting. Isabel did a very nice presentation about all the different methods and if they were suited for treated seeds or not. Because depending on the method, you can have a result or you can have an interference from the treatment with the result. So maybe that's something where we could go in the future, just in our methods, to give some indication, that not that they are validated, because we will never be able to say it, but they are suitable for this or this sort of method of treatment, and not for the others. But for the moment, that's true that we don't have methods for that. What we do in my lab, and I know that in some other labs there are some also, when we have treated seeds, generally we add extra controls, just to be sure that the treatment does not influence the method and the result of the method. And another question: hope the Danish Institute for Seed Pathology for Developing Countries is actively involved in ISTA's SIDELS testing efforts. Please clarify. I don't know. We had a member of Denmark a few years ago, but not anymore. Guro, do you know more?
I just wonder if at least this Danish DJISP, it was called, it's merged with the University of Copenhagen at least. It's not own organization any longer, as far as I know. It's been merged with the university.
Okay.
But I have no idea. I don't know anyone working on what they are doing any longer. I used to know them quite well, but not now.
Okay, and me neither. Sorry. Yeah. Okay, and we looked at all the questions. So I want to thank all the panelists for their very nice presentations, and all the work which is behind the presentation of all the research and validation work. Thanks a lot to Andrea, who helped us today to change between presenter and so on, which is not always easy. So thanks a lot for staying with us, Andrea. And thanks a lot to you all for attending this meeting. We were, depending on the moment, around 70 people, so from all over the world. So, well, thanks a lot for your participation. And now I wish you a, well, good day, good evening, good night, because I know that for some of you it's the night now. And thank you for your participation. Bye-bye.
Bye.